The small green leaves and white flowers belong to the Moringa oleifera tree, known throughout India as the drumstick tree. For centuries, it has been a staple of local nutrition. Families have used the leaves, pods, and seeds in daily meals, recording their impact on health through generations of observation. This history is preserved in the ancient Indian medical system of Ayurveda. Traditional records on palm leaf manuscripts describe using the plant to address stomach ailments and joint pain, providing modern researchers with a historical map of its potential effects. Today, this ancient record has met a global market. Moringa is now widely processed and sold as a modern superfood, often accompanied by claims that it can solve a wide range of modern health problems. While these historical accounts provide a starting point for research, they are not scientific proof. To find the truth, the plant must move through a sequence of modern biological tests. The first level of testing begins with chemical extraction. Scientists take the physical leaf and break it down to identify every individual molecule hidden inside the plant tissue. Advanced laboratory fingerprinting reveals these leaves contain exactly 79 distinct bioactive compounds, ranging from protective antioxidants to antibacterial acids. Among these are proteins and complex fiber chains called polysaccharides, which travel deep into the digestive system intact. The raw chemical data confirms that the leaf provides a concentrated supply of the exact molecules the human body uses to regulate its internal systems. In the second tier of the gauntlet, researchers move the extracted fibers into a controlled environment to observe how they interact with living cells. When beneficial gut microbes like Bifidobacterium longum encounter these fibers, they begin to ferment them. As the bacteria consume the complex plant sugars, they rapidly multiply and release new beneficial compounds into their environment. These molecules signal the intestinal wall to begin repairs. They trigger the production of tight junction proteins, which act as physical gatekeepers that seal the microscopic gaps between cells. Under these isolated conditions, the plant's components successfully trigger the biological process required to repair the gut barrier. The testing now scales up to living systems. In this stage, the molecules must survive the entire digestive process and interact with a functioning bloodstream and organs. During a 12-week animal trial, mice on a high-fat diet were supplemented with moringa leaves. The results showed a significant reduction in weight gain and a decrease in stored fat. Researchers traced this effect to a liver genetic switch called SREBP1C. The leaf compounds turn this switch down, slowing fatty acid synthesis. This shift also quieted inflammatory alarms, significantly lowering levels of inflammatory proteins circulating in the blood. In these standardized animal models, the plant's active components demonstrated the ability to restore microbial balance and calm systemic inflammation. The final and most difficult stage of the gauntlet is the human clinical trial. This is where laboratory success meets the reality of human biology. Unlike identical lab mice, human bodies are defined by diversity. Differences in our genetics, our diets, and our environments mean that the same molecule can produce very different results from one person to the next. This leads to conflicting data. While one trial of postmenopausal women showed a 15% drop in blood sugar, a separate study of diabetic patients showed no significant change in their long-term sugar markers, known as HbA1c. These results show that the biological mechanisms we observe in the lab do not always translate into universal clinical outcomes for every human. This variance in human trials doesn't mean the plant has failed the test. It provides a more realistic understanding of how it works. While it may not be an overnight medical cure, the data confirms it is a remarkably safe and exceptionally nutrient-dense food. Its high concentration of proteins, fibers, and antioxidants offers reliable daily support for the maintenance of a healthy microbiome. By filtering ancient tradition through rigorous scientific testing, we find a plant that offers genuine biological value without the need for exaggerated marketing claims.